Sexual reproduction in flowering plants. Exercises. Name the parts of an angiosperm flower in which development of male and female gametophyte takes place. Here, male gametophyte means they are pollen grain because pollen grain contains gametes. Female gametophyte means embryo sac, and embryo sac is present inside ovule and pollen grain is present inside anther.
Differentiate between microsporogenesis and megasporogenesis. Which type of cell division occurs during these events? Name the structures formed at the end of these two events. Microsporogenesis means it involves the formation of microspores from microspore mother cell, and microspores will later become pollen grains. Whereas megasporogenesis means the formation of megaspores from megaspore mother cell. Now one of the megaspores will become functional and later forms the embryo sac. So in which the pollen grains will be the male gametophyte and embryo sac will be the female gametophyte. Now for the second part of the question, that is, which type of cell division occurs during these events? So if the microspore mother cell has to become microspore, the type of cell division is meiosis. Similarly, if the megaspore mother cell has to get converted to megaspores, then the type of division is meiosis. Because it's a reduction division, megaspore mother cell is diploid and megaspores are haploid. Similarly, microspore mother cells are also diploid, and when it has to become microspore, that is haploid. So it is a reduction division, meiosis. And then comes the question, name the structures formed at the end of these two events. So at the end of these two events, it forms the pollen grains and embryo sac. Arrange the following terms in the correct developmental sequence. Pollen grain, sporogenous tissue, microspore tetrad, pollen mother cell, male gametes. Here there should be sporogenous tissue which is found inside the anther. Now suppose this is the transverse section of anther. There will be microsporangium in each lobe. So at the center of the microsporangium there is the sporogenous tissue. And it's within this sporogenous tissue that there will be the pollen mother cell or microspore mother cell. And they are diploid. The pollen mother cell undergoes meiosis to form a microspore tetrad. This is a microspore tetrad. Later, each of this microspore will separate and they will mature to form pollen grains or microspores. So each one of them will become microspore and they will be haploid. So each microspore will later mature to form pollen grain. And within the pollen grain, there will be two male gametes. So gametes will be present inside the pollen grain. So pollen grains are actually male gametophyte, which means they are the carriers of male gametes. Now to form the order, so the first will be sporogenous tissue, and inside the tissue there will be pollen mother cell. Then pollen mother cell will divide to form microspore tetrad, which will become the pollen grain, and pollen grains are the carriers of male gametes. So this will be the sequence. With a neat label diagram, describe the parts of a typical angiosperm ovule. So this is a ovule, and ovule has got protective integuments, means protective coverings. Excepting one opening, which is called as micropyle. So they are integuments. And there is a stalk that connects the ovule with the ovary wall over here. This stalk is called as funicle. And then the tissue which is present within the ovule, this tissue is called nucellus. And here every cell will be diploid. Even the ovule is diploid. So there is nucellus present within the ovule. So this is nucellus. And then later on, near the micropylar area, there will be the megaspore mother cell, which undergoes cell division and becomes megaspores, of which one will become functional, leading to the formation of embryo sac. And since embryo sac is formed from megaspore, that's why embryo sac will be haploid. And this will be micropylar end, the opposite end of ovule is called as chalazal end. What is meant by monosporic development of female gametophyte? So female gametophyte means it is the embryo sac. How is embryo sac formed? Embryo sac is formed from a functional megaspore. But there were, before this functional megaspores, there were 4 megaspores. So there were these 4 megaspores, haploid megaspores, that developed from megaspore mother cell, which was diploid. So megaspore mother cell underwent meiosis to form the 4 megaspores, of which 3 got degenerated. After the degeneration of these 3, only 1 functional megaspore remained that developed into embryo sac. So that's why monosporic development means it is a development of 1 functional megaspore from megaspore mother cell. A neat diagram explains the 7-celled 8-nucleate nature of the female gametophyte. In this, the female gametophyte means embryo sac. So they are actually asking to draw the diagram of embryo sac. Now suppose this is the embryo sac. So there would be one end called the micropylar end. And the opposite end is called chalazal end. At the chalazal end there would be 3 cells. And these 3 cells are called antipodal cells. Then there will be a central cell. And central cell has got 2 nuclei, but they are not fused together, they are separate. And these nuclei are called polar nuclei. And then at the micropylar end also there are 3 cells. These 3 cells are together called as egg apparatus. In which the center cell is the egg cell. The other 2 cells which are there here, this and this, the other 2 cells are called synergids. And the synergids have got special finger-like projections like this, which is called as Filiform Apparatus. And Filiform Apparatus is for guiding the pollen tube towards the synergids. And therefore you can see there are 7 cells, 3 antipodal cells, 1 central cell and 3 cells inside the egg apparatus at the micropylar end. So totally there are 7 cells, but if you see nuclei, there are 8 nuclei. That is 1, 2, 3, 3 are there in these antipodal cells, 2 in the center, and then 3 nuclei are there here in the egg apparatus place. What are Chasmogamas flowers? Can cross-pollination occur in Clestrogamas flowers? Give reasons for your answer. Mention 2 strategies evolved to prevent self-pollination in flowers. So we have learned the outbreeding devices. In that 3-4 methods are there, in which any 2 strategies we have to answer here. Now Chasmogamas flowers are open flowers. So the petals are open like this, and so there are chances that pollen grains from the neighboring flowers can come and fall onto the stigma of this flower. So cross-pollination can happen in Chasmogamas flowers. Now about Clestrogamas flowers. Clestrogamas flowers are closed. Their petals are closed. So since they are closed, there is no chance that a foreign pollen grain can come and fall in the stigma of a flower. So this is, suppose if it is Clestrogamas, means the flower will be completely closed. So that's why always self-pollination only will occur in case of Clestrogamas flowers. Cross-pollination will not happen in Clestrogamas flowers. as here they are closed flowers, petals are closed. Now, since continuous self-pollination can result in inbreeding depression, the plants also have adopted the strategies to prevent self-pollination. So some of them are like this. In some species, pollen release and stigma receptivity are not synchronized, means they both are not at the same time. So either the pollen grain is released before the stigma becomes receptive in the flower, or stigma will become receptive before the pollen grain is released. So this can prevent self-pollination. And some other species, the anther and the stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. So that would be somewhat like this. Suppose this is the stigma of a flower. Anther can be in another position, like this. If that's the case, these anthers, it will be difficult for these anthers to fuse with the stigma within the same flower. Thus, self-pollination can be prevented by location. The position is different. Stigmas and stamens positions are different. Now, both these devices can prevent autogamy, that is self-pollination. Now, there's a third device to prevent inbreeding, and that's called self-incompatibility. This is a genetic mechanism. It prevents self-pollen, that is pollen from the same flower, from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Now here, this anther and the stigma both are belonging to the same flower, but this stigma has adopted self-incompatibility. Which means that stigma will not accept the pollen grain from its own flower. What is self-incompatibility? Why does self-pollination not lead to seed formation in self-incompatible species? Self-incompatibility means when the stigma doesn't accept self-pollen to come and germinate. So there is no self-pollination possible in such kind of flowers, and therefore it will not lead to seed formation. What is bagging technique? How is it useful in a plant breeding program? The bagging technique is used during artificial hybridization, where suppose there is flower. Suppose this is flower number 1 and there is another flower. Flower 2. Now we want the pollen grain from the flower 2 to get tested on the stigma of flower 1 to get a hybrid, but we don't want self-pollination. So for that purpose, the anther and the stamens of the first flower will be removed, and that process is called emasculation. After the emasculation, now this emasculation is done when the flower was in a bud stage. So the stigma has to become receptive to do the hybridization. So after the emasculation, they will be bagged with butter paper, and that process is called bagging. So that the cross-pollination can be prevented. Then once the bud becomes receptive, or the stigma starts to become receptive, the bag can be removed. And the desired pollen grain, whatever desired pollen grains are there, desired pollen grain can be dusted from the desired flower to the stigma, and then it will be rebagged. They are rebagged because there should not be chances that again some cross pollen comes and falls in this flower stigma. That's why they are rebagged. And then the fruits will develop inside this bag, and those fruits will be our required hybrid. So that's how it is useful in plant breeding program. What is triple fusion? Where and how does it take place? Name the nuclei involved in triple fusion. Now, triple fusion is like this, where there will be two male gametes inside the pollen tube. In that, one male gamete will fuse with the egg, but the second male gamete will enter into the embryo sac and they will fuse with the central cell. Central cell has got already two nuclei. So when the male gamete also joins, there will be three nuclei. They will fuse, and that's why it's called triple fusion. And so it takes place within the embryo sac. And the nuclei that is involved in triple fusion are one polar nuclei and then nuclei of male gamete. Why do you think the zygote is dormant for some time in a fertilized ovule? Because zygote has to derive its nourishment from the endosperm. That's why they will be dormant. Differentiate between hypocotyle and epicotyle. Hypocotyle is a tissue that comes, there are two cotyledons like this. In case of a dicote embryo, and this is the embryo axis. Part of the embryonal axis which is just above the level of attachment of the cotyledons. So these are cotyledons just above the level of attachment of the cotyledon. This part is called epicotyle tissue that will later form the shoot. They will form the plumule and the shoot of the plant. And this is the level of attachment of cotyledons. And the part of the tissue below the level of attachment of the cotyledons, that tissue is called hypocotyle tissue. And that will terminate into this part, that is radicle, which will later form the root. So this will be epicotyle, and that will terminate to form the plumule or shoot of the plant. And these both are cotyledons. And the same happens in monocot seeds also. Only thing is that the monocot seeds cotyledons are called scutellum. Differentiate between coleoptile and coleorhiza. Now if it is a monocot seed where there is the scutellum, this is a scutellum, that is the cotyledon of monocot seed, like maize. So this is the level of attachment of cotyledon. And the tissue which is there here, they will be called as epicotyle tissue. And below the tissue is called hypocotyle. So the epicotyle tissue will develop to form the plumule part. and wherever the plumule is present, it's going to be the shoot of the plant. So above the plumule, what protective coatings are there, they are called as coleoptile. And the coverings for the radicle that later becomes the root of the plant, those coverings are called coleorhiza. So coleoptile is the protective covering for the shoot of the monocot seed, and coleorhiza is the protective covering for the radical part of monocot embryo. They differentiate between integument and testa. Now in case of integuments are protective covering for ovules, whereas ovules after fertilization becomes seed, and so the same integument will be called as testa. So testa will be the covering for seed. Seed what covering comes, that is called as testa, which was once upon a time integument when the seed was called as ovule. Before fertilization, seed was called as ovule and testa was called as integument. And the difference between perisperm and pericarp. Pericarp is the ovary wall. In a pistil, there is the ovary wall which is present. This is called a pericarp. In other words, it's the fruit wall because ovary becomes a fruit after fertilization. And perisperm means in seeds like beet or pepper, black pepper. The tissue which is present inside the ovule, which is called as nucellus, and it's from the nucellus that ultimately embryo sac is made. But still inside the embryo sac or inside the seed, some remnants of nucellus will be present, which happens in case of seeds like beet and pepper. Nucellus will be present inside the mature seeds also. Then they are called as perisperm. Why is apple called a false fruit? Which part of the flower forms the fruit? Now usually the ovary, when the ovary becomes the fruit, like in mango, ovary becomes the fruit. Then they are called true fruits. Below the ovary, there is a tissue which is called as thalamus. This tissue is called thalamus. In case of apple, the fleshy sweet part which we are having, that is actually from thalamus. And if you closely see, you can see the ovary present here in apple with seeds here. So it's not the ovary which we are feeding on, we are actually eating the thalamus part. So this becomes a false fruit, because true fruit means it should develop from ovary. What is meant by emasculation? When and why does a plant breeder employ this technique? Emasculation means to remove the stamens from the flower. Why the plant breeder is using? When he wants to do artificial hybridization. Because he doesn't want the stamens of the same flower. He wants the pollen grain from another flower to get deposited on stigma. So that's why plant breeder does the emasculation. which means removal of stamens from a bisexual flower. If one can induce parthenocarpy through the application of growth substance, which fruits would you select to induce parthenocarpy and why? Means whichever fruits you don't want the seeds, those fruits you can induce parthenocarpy. Like it can be watermelon, lemon, orange, grapes, which you would like to eat without seeds. That you can use the hormones and growth hormones and you can make it seedless. Explain the role of tapetum in the formation of pollen grain wall. Now, what is tapetum? If this is the anther has got walls, like the outer wall is called ectoderm, and the next wall layer is called endothecium, and there are middle layers over here which are also walls, and there is a layer called tapetum. Now, tapetum provides nourishment to the developing pollen grains. So if there are any pollen grains present over here, so the pollen grains have to develop walls, and so tapetum has to nourish these pollen grains. So that's the reason why tapetum is there. Now, what is apomixes and what is its importance? Now, apomixes means when the seeds are produced without fertilization, they are called apomixes. And its importance is that there will not be any segregation of characters once it is made as an apomictic seed. So that's why once we make a hybrid seed, and then if you convert them into apomictic seed, then it will not lose its hybrid nature. Therefore, every generation there is no need to buy hybrid seeds once it is made into apomix.